Normal crystalline lens is a transparent biconvex structure located posterior to the iris and anterior to the vitreous body. The lens is suspended by numerous fibers that together are called the zonule. Collectively, this ring of fibers, zonule of Zinn, attaches the lens to the ciliary body and can be considered a ligament. Components of the lens include the capsule, the epithelium, the cortex and the nucleus. This is the capsule, epithelium, cortex, nucleus. An imaginary line called the optic axis joins the anterior and posterior pole of the lens, passing through them. Hypothetical line on the lens surface that pass from the one pole to the other are referred as meridians. The equator of the lens is its greatest circumference. The function of the lens to maintain its own clarity, to refract light, to provide accommodation in conjunction with the zonule and the ciliary body. Lacking a blood supply and innervation after fetal development, the lens depends entirely on the aqueous humor to meet its metabolic requirements and also to remove its waste. The lens is able to refract light because its index of refraction, normally about 1.4 in the center, 1.36 in the periphery, is different from that of the aqueous and vitreous humors surrounding it. In its non-accommodative state, the lens contributes approximately 20 diopter of the approximately 60 diopter of convergent refractive power of the average human eye. The ear-cornea interface provides the rest, about 40 to 45 diopter. The lens continues to grow throughout in individual life. At birth, it measures about 6.4 mm equatorially and 3.5 mm anterior posteriorly, and weighs about 90 mg. The lens of an adult typically measures 9 to 10 mm equatorially, about 5 mm anterior posteriorly. Weighs about 255 mg. With increasing age, the relative thickness of the cortex increases. The lens also adopts an increasingly curved shape so that older lenses have more refractive power. However, the index of refraction of the lens decreases with increasing age, probably as a result of the increased presence of insoluble protein particles. Thus, with increasing age, the eye may become either more hyperopic or more myopic depending on the balance of these opposing changes. Now the lens capsule. It is elastic, transparent basement membrane that is composed of type 4 collagen and other matrix proteins and laid down by epithelial cells. So this is made of type 4 collagen. Capsule contains the lens substance and is capable of molding it during accommodative changes. The outer layer of the lens capsule, the zonular lamella, also serves as the point of attachment of the zonular fibers. The lens capsule is thickest in the anterior and posterior pre-equatorial zones and thinnest at the central posterior zone. At birth, the anterior lens capsule is considerably thicker than the posterior capsule. Its thickness increases throughout a person's life. Zonular fibers. As mentioned, the lens is supported by a system of fibers, the zonules, that originate from the basal lamina of the non-pigment epithelium of the pars plana and pars plicata of the ciliary body. These zonular fibers, which are located in the valleys between the ciliary processes, consist of microfibrils composed of elastic tissue. They insert at discrete points on the lens capsule 1.5 mm anterior to the equator and 1.25 mm posterior to the equator. With increasing age, the equatorial zonular fibers regress, leaving separate anterior and posterior layers that appears in a triangular shape on a cross section of the zonular ring. Fibers are 5 to 30 micrometer in diameter. On light microscopy, they are revealed in eosinophilic structure that have a positive periodic acid-skip reaction. Immediate posterior to the anterior lens capsule is a single layer of epithelial cells. These cells are metabolically active and carry out all normal cell activities, including biosynthesis of DNA, RNA, protein, lipid. They also generate adenosine triphosphate to meet the energy demands of the lens. The epithelial cells are mitotic. The greatest activity of pre-mitotic DNA synthesis occurs in a ring around the anterior lens known as the germinative zone. The newly formed cells migrate towards the equator, where they differentiate into fibers. This area called the bow region is where the epithelial cells begin the process of terminal differentiations into lens fibers. This is bow region. During this differentiation, perhaps the most dramatic morphologic change occurs when the epithelial cells elongate to form lens fiber cells. This elongation is associated with a tremendous increase in the mass of the cellular protein in the fiber cell membrane. At the same time, the cells lose organelles including nuclei, mitochondria, ribosomes. The loss of these organelles is optically advantageous because light passing through the lens is no longer absorbed or scattered by these structures. However, these new lens fiber cells lack the metabolic function previously carried out by organelles. They are now dependent on glycolysis for energy production. Nucleus and cortex. As new fibers are laid down, no cells are lost from the lens. The new fibers crowd and compact the previously formed fibers. The older layers are located toward the center. The oldest layer, the embryonic and fetal lens nuclei. They were produced during embryogenesis and persist in the center of the lens. The outermost fibers are the most recently formed and make up the cortex of the lens. Lens sutures are formed by the interdigitation of the anterior and posterior tips of the spindle-shaped fibers. Multiple optical zones as well as Y-shaped sutures located within the lens nucleus are visible on slit lamp